without further ado, want to switch it right over to our main presenter today, Michelle Boone. We're very excited to have her here with us. She actually specifically asked me not to tell you a lot about her because she's going to go ahead and do that on her part. But the thing that she did want to hit home is that she is a new member of the Native Bee Society, and she's very excited to be a member. We're super excited to have her and all of you as members as well, because it really takes all of us and our brain power and thinking. So we're really excited to have her, and especially today presenting. So without further ado, I am going to switch it over to Michelle so she can begin presenting.
Great, thank you. Can folks hear me okay?
Yes.
I can hear you. Yeah.
Awesome. So I do want to mention before I get started, I am having some bandwidth issues here. So I'm going to turn my camera off once I start talking. And I did get kicked off of the meeting a couple times here or during these last half hours. So if that happens, it seems to come back pretty quickly. So if I lose connection, don't worry, I'm coming back. So with that, I'm going to go ahead and share my slides. Great. All right.
Michelle, do you want to turn your video off?
Oh, did I not do that? Sorry, I'm I'm on my work computer, which uses Teams, and something about the Zoom platform is confusing me here. All right, let me figure this out. There we go. All right, can folks see my screen?
Yes.
I can see your screen, yes.
But I think the screen we're seeing is your presenter screen as opposed to the public screen if you have two options.
Ooh.
We can see your notes.
I see that. I didn't realize I had it like that.
Isn't it a
Still
button
works
you
just
switch
fine.
to? Use presenter view, click that off maybe? Does anybody else know? I just know people help me do it. I don't know how.
I think she just needs to click from the
If you
beginning.
click on setup slideshow, Michelle, click on setup slideshow, and then it should give you options for how you want to present it, whether you want it presented as a full screen or whether you want it presented in the window. There's nothing happening when you click on that.
Oh, I see it, but which should I pick, full screen or window?
I would pick window.
Window, okay. All right, now can people see the right view? Great.
Yes.
Yeah.
Awesome. Okay. Thank you for your patience. And I'm going to introduce myself, but I got so excited when Joe G shared a picture of a Bombus affinis and then asked about or commented about wondering where the nest is. So while we were doing the photo slideshow, I was like quickly throwing together some slides. So I'm going to show you those, and then I'm going to do my actual introduction. So the question was, well, where would those rusty patch bumblebees be nesting, in this residential area? And so I did my PhD work, my dissertation work, which I'm going to share today in Minnesota, and I focused on rusty patch bumblebee. And while I was there, I had the opportunity to engage in some nest removals and some nest studies. So I wanted to share some pictures. So most of the time, bumblebees are going to be underground, but they live generally in abandoned rodent holes, but these things can be long. So I just wanted to share this series of photos. On the furthest left, you can see we just arrived at this house, and we know the entrance is, in that little dirt patch. And so we use baby powder and we blow it through the hole to kind of keep track of the tunnel as we started to dig. So then the second picture is showing, okay, we're digging down, we're following this tunnel. And then the last picture is, I actually think we hadn't quite found the tunnel yet, or the nest yet, but you can see how long and winding the tunnel is getting. So that's one place where bumblebees might be nesting, including those rusty patch bumblebees you saw, Joe. And also in 2020, we did a study specifically of rusty patch bumblebee nests, an opportunistic study because there were a couple nests reported. So these are two locations where rusty patch bumblebee nests were reported in Minnesota in urban areas. So the one on the left is someone's garden, and that's extremely shallow, still probably an abandoned rodent's nest. But all we've done here, we didn't even dig, we just pulled back the landscape fabric and the nest was right there. And you can see there's even some garbage in there, some plastic. And then on the right was a more unusual nest location. So that's actually in the foundation of someone's house. So the top is like, imagine you're going under someone's house, actually under the foundation. And that's kind of this ledge where there was some insulation. So the picture at the top is where the nest was after we removed it. It was really hard to get a picture of the nest in place. But then the bottom picture is the nest that we had scooped out of there. And you can see the foundation from the walls in the nest. And we collected these nests after the colony cycle and of course had to get special permits from Fish and Wildlife Service. Anyhow, that's not what this talk is about. I just threw those slides in, but I got really excited. So let me backtrack and start with my introduction. So it's great to meet you. And as was mentioned, I'm a fairly new member of the Washington Native Bee Society. I was born and raised in the Midwest in Minnesota, and that's why this talk is about bees in Minnesota and not bees in Washington. So I appreciate folks still showing up, even though this isn't a local topic. So I completed my PhD in entomology pretty recently in 2023, so I'm pretty freshly minted. I moved to the PNW. I actually live in Portland, Oregon, in 2022. So before I finished my dissertation, I really wanted to live out here. So I decided to make the move first, try to do some networking and see what happens. And so far it's worked out. And so that picture is an event. I just like that picture, so I wanted to share it. But I'm a Xerces volunteer ambassador, and I think some other members of this society are as well. And then I also joined the Washington Bee Atlas and the Native Washington Bee Society as well when I got here to learn more about local species and local conservation issues. When I first graduated, I was a postdoc at Washington State University. So I did work in Washington for a little bit, but I didn't do any bee work there. Otherwise, I would have talked about that instead. But I did do some work on Western monarchs out in Eastern Oregon and Washington. And I just threw some pictures on there for fun. But essentially, we set up a field study where we had put these cages over 2 different kinds of milkweeds and in sun and shaded environments. And then we put monarch larvae into these enclosures and tracked various survival metrics across the season. But that's for another day. And then currently I'm a contractor with the National Park Service. I'm a pollinator project manager with the inventories program. And I oversee inventory projects in various parks across the country. We're Washington based. But technically I'm hired through the Natural Heritage Program of Colorado. So kind of a winding path. And through all of it, I haven't actually done any bee work in Washington. So that's why you've got the topic you have today.
Michelle, we do have one question.
So I just wanted to share this. Oh, yes.
Just one quick question. Somebody was interested in knowing where in Minnesota you're from. They're from Bird Island.
Oh, I am not familiar with that. I'm from Minneapolis.
Awesome. Cool. Thank you.
Yeah, of course. And yeah, so I can't see you. So yes, please, if anyone has questions, just shout them out. So this map it's really just for, again, just for fun. I'm not going to talk about my work with NPS tonight. But this is a map of our current pollinator inventory projects that are either slated to start this summer or next summer, and these go through 2026. So again, we haven't had any requests from parks from Washington, so we don't have any current projects that would be pertinent. So that's kind of my path into bees. Now we're going to try this Mentimeter activity, and I'll admit I have mixed success with these working, but let's see if we can get it to work. Ooh, here it comes. Okay, so if you haven't done these before, you can use your phone or your computer. I find sometimes if I'm watching a presentation, it's easier on my phone, but you go to this website, menti.com, and then it should ask you for a code, and you'll type in this code, and then you can answer this question. And it's going to make a word cloud. So
I'm just seeing your screen, your PowerPoint.
oh, you can't see it. Okay, you're not seeing what I'm seeing. I was worried about that. But I'm determined to make this work. So let's see what I can do.
Hey, Michelle, I just Googled menti.com and got a web page that says enter the code. So if others do that, we can get to it pretty quickly.
Oh, well, let me then see if I can just put the code in the chat, since I'm guessing you can't see it on the screen. Okay. So you'll go to menti.com and you'll enter the code, which I'm about to put in the chat. Sorry, this is clunky. This is why I usually don't use it. Huh, that's not working. So the code is here. We'll give this one more minute, and if I can't get it going, we'll move on. But it's not letting me copy and paste the code.
Can you speak the code?
I'm typing it right now. It's 3276926. And the question is, what got you interested in bees? But try to keep it to like 1 or 2 words, because it's going to make a word cloud. So like a whole phrase won't make as pretty of a word cloud.
I'm sorry, 3276.
It's 3276926. And I typed it in the chat.
Thank you.
Of course. What got you interested in bees? Okay, stuff is coming in. I can see things, and I'll give people like another 30 seconds, and then I will just copy and paste our word cloud right into my PowerPoint so everyone can see it. Thanks for your patience.
Yeah, we're getting a lot of responses, 50, 55, 58. That's awesome. People are doing it. That is so cool.
All right, let me go back and share my slides again.
I think if you select share your screen instead of share your window like you did at the beginning, you might be able to see what you're seeing regardless of
What I'm doing.
what you're looking at. Yeah.
Thank you. See, I don't know what I'm doing. You would think by now, I mean, we've been in this virtual world for 4 years, and I still can't quite figure it out. But here we are. All right, now can people see my slides with a word cloud on it?
Yes.
Yes.
Yes. Perfect.
Good job.
Okay. A little bit clunkier than I thought, and I think it got a little bit cut off there, but we've at least got 55 responses. And if you've never made a word cloud, the size of the word indicates how many people selected that. So photography is a big one, gardening, entomology, lots of plant related stuff. I see Karen right on there. That's funny. We've got hiking. So lots of folks. I'm getting the sense we have a lot of gardeners, a lot of nature folks, excuse me, some connections between birds. So that's great. That's awesome. And actually, similarly, before I started grad school, and after I had a gap year, I worked as a public gardener, and that's what actually got me looking at pollinators and really fascinated by them. So great, thank you. I appreciate that, and it helps me to know my audience. And I see childhood in Minnesota on there. That's great. All right, and so I was maybe because I'm just so used to hanging around with entomologists. I was expecting people were going to put like insect decline or like bee apocalypse or something in there. So then that was going to be my smooth transition into this slide. But regardless, the reason why there are so many folks that are getting into entomology to help study pollinators is really driven by insect declines. And so insect declines have been documented globally, but it is really important to remember that sampling effort is really uneven. Oops, I didn't quite finish this slide. So the geographic distribution of sampling, it's pretty concentrated, primarily in more developed countries. So the US and Europe is where the bulk of this work has been done. But even within those areas, it's concentrated, in areas where people are. So there's a lot more data for, say, like coastal areas or around cities than there are kind of out in larger expanses because, it's logistically challenging to get out there. There's just fewer people out looking. So we have to, look at decline data, with a grain of salt, that it's really not complete. And I was going to put the number of described insect species that have been studied or monitored, but I'll admit it's been a hectic week and I never quite got around to that. But we will say it is very, very, very few insect species that have been described have actually been studied or monitored in any sort of significant way. Regardless, that doesn't mean we shouldn't do anything or shouldn't care. And learning why these declines are happening and then finding ways to halt and reverse declines of beneficial insects is really important for our food supply, as well as the stability of terrestrial ecosystems. And popular media, despite it can certainly have benefits and has helped raise awareness, but has also played a role in really complicating the issue of really bee declines in particular. And I know this group is educated and knowledgeable about bees, so I don't need to do my spiel about how honeybees aren't native, but they are even now, even with the listing of rusty patch bumblebee. Most people do still think of bees as honeybees, and when you think of the Save the Bee movement and those types of conservation messages, they are still largely centered around honeybees. And so I just grabbed this infographic of the hundreds that I could have that illustrate this issue. So they're kind of mixing their facts and their interpretation a bit. So yes, there's data to support that around 30% of our world's crops, and this isn't even really accurate, are pollinated. They're all pollinated. More accurate would be to say insect pollinated. And really even more accurate would be to say 30% of world crops depend on insect pollination. But there are many more crops that experience higher yields with insect pollination. And the 90% of our wild plants are pollinated by insect pollinators is also fact-based, but then they throw in this 40% of, what does that say, 40% amount of bees being lost from keepers. So now it's kind of bringing in this issue of honeybee colony collapse, which really isn't a conservation issue at all. It's, as you know, it's an agricultural issue. So there's a lot of this messaging going on. So I really appreciate groups like the Washington Native Bee Society that are, and Xerxes Society, and lots of great groups out there that are really helping with public education around these issues. So bumblebees, specifically of the genus Bombus, have declines that are well documented. So these are one of those species that are better studied. And likely because they're large, charismatic, I'm sure I just pronounced that wrong, easy to detect species, similar like the monarch. They're sort of these flagship species. But because of that, we actually do have pretty good decline data. Some of our longest data sets actually come from the EU. And starting back in the 70s, Paul Williams, a famous bee researcher there, had already started to note declines happening in the UK. So we do have quite a bit of data and some long-term data sets for some species, I should say, showing these declines in bumblebees. So it is really important that we start learning more about them, but even more importantly, start taking action to reverse those declines. And you may have heard this statistic. So around a quarter of bumblebee species globally that have been assessed are either vulnerable, endangered, or critically endangered, which essentially means they're in decline. And so just a little bit of bumblebee background. And I was so excited to see all of the bumblebee images being shared, and now we can learn a little more about them. So globally, there's about 265 species of bumblebees. In North America, there's around 48. And you have more species out here in Washington than we do in Minnesota. In Minnesota, we've got just about half, but you actually have more than half of the known species in North America in Washington. And I actually just had to update this slide today because I had looked at an old source that said there were 25 species in Washington, and then learned in a presentation from Karen Wright today that it was 36. So I had to quick go in and update my slides so I wouldn't be embarrassed. So there are currently 2 species listed as federally endangered in the US. The rusty patch bumblebee, Bombus affinis, was the first in 2017. Bombus franklini, I think it was 2021, it is a Western species, but it's very limited in geographic range. It's found in the southern part of Oregon and northern California. So not quite in Washington, but much closer than the rusty patch. It's presumed largely to be extinct or possibly extinct with the last sighting in 2006. That said, there's a lot of land and land that's hard to access, like high elevations, or it's just hard to do surveys, so it could still be out there. Additionally, there are 6 more species that are currently have been petitioned, and they're all at different stages of the process. Some have already undergone their species status assessment, some are in progress, some have just been listed. But I wanted to show them here. And we're going to talk more later about the Western bumblebee, particularly because it can be found in Washington, and I wanted to at least talk about one local species. And that one, it was petitioned back in 2015, and just to give you an idea of how long these things take, the listing decision is expected in 2024. It was actually originally expected in 2023, so it's even been pushed back. And then there's one species that has been petitioned. It was assessed. It went through that process, but the Fish and Wildlife Service determined that listing wasn't warranted, and that's Bombus terricola. So that was a lot of background. I'm actually going to talk primarily about rusty patch bumblebee today, so I'm glad that it already came up. So as I mentioned, this was the first bee species listed as federally endangered in the continental US. And that continental piece is important because there were several species, bee species, not bumblebees, in Hawaii that were listed first. So I had a couple pictures here just to show you the color pattern. So on the left, that's a worker. That's a worker and male pattern, but that's a worker. And you can see where it gets its name, the rust-colored patch on the second tergal segment. an important distinction between rusty patch and say like a red belt or Bombus griseoculus. I'm really bad with common names. Brown belted bumblebee. Um, Note for one, the shape, it's kind of like a W shape where the color is. And then also really important is that there has to be kind of a yellow band under the rusty patch. And then another key identification on at least the males and workers is this thumbtack shaped black spot in between the wings. The queens are a lot harder to identify. They don't have the rusty patch. They don't have the thumbtack. They just have a kind of nondescript black dot with the first two tergal segments yellow. They are kind of have like a round shape, and that's also kind of helpful. Round shape and close cut hair, which can be used to differentiate them from, say, like a Bombus vagans, which is also a similar color pattern in the region. So here's the historic range of rusty patch bumblebee. It's essentially just ignore all of the X's and dots. Just look at the gray outline. Someone had asked in the chat earlier if they're found out west. They're not, and they never were. It's an eastern species. It also used to extend northward into Canada. It doesn't, stop along the border of the countries. But here's the known extant populations within that historic range. So most of the known population now that's hanging on and seems to be at least relatively stable for now is in the Great Lakes. So in Minnesota, we have, at least in sort of the southeastern part of Minnesota, we do have a fairly robust population that extends across Wisconsin into Illinois and Iowa. But excitingly, there's also this Appalachian population. And when I started grad school in 2017, this map didn't show anything east of Ohio. So those are recent sightings. So there is a lot of work being done, which was, of course, sparked by the listing. So we're learning more and more about this species. And so monitoring of bees is really important, especially as we have all of these species being petitioned, we have species being listed. It's important to monitor bees. And of course, that's important for tracking declines. But importantly, I would even say even more importantly is for assessing how our land management actions and conservation activities affect bee populations. I always say monitoring for the sake of monitoring, if you're not going to do anything about it, is kind of a waste of your time and effort. So if you see a decline, you do need to do something and then continue that monitoring to see how you're doing, to see if the changes you're making are actually positively influencing the population. And a key part of that, and the topic of this talk, is that when you're doing these sampling and monitoring activities, it's really important to account for detection probability. It's important for designing your surveys, but also it's crucial for interpreting monitoring data. And don't worry, we're going to define detection probability in a couple slides here. But first, I wanted to do a little thought experiment to get us in the mindset of detection probability. So I want you to answer this question to yourself, or you can put it in the chat if you want. Are there any dogs in the room with you? So this is just a simple presence absence question. And you might think, well, that's silly. Of course, you know if there is or not. But how likely do you think, to find all the dogs in your room if there are, in fact, any dogs there at all? Again, a silly question. Unless you have a really sneaky dog, you're going to be able to find it pretty easily. But let's think about those same questions, but this time let's think of spiders. So are there any spiders in the room with you currently? This is, again, a presence absence question, but it's going to be a lot harder to answer. You're going to need some time to search. And if we ask you the next question, how likely are you to find all the spiders in the room? I think most people, unless you just really, really are good at searching all the nooks and crannies, you're not going to be 100% sure that you found all the spiders in the room. You just can't. And the same applies to bees. So, if you're out, you're in your garden or you're out at, a park or wherever, how likely are you to find all the bees at that site if indeed there are any bees? And of course, you're not going to find them all. We can't find them all. Sometimes they're just not going to be available for detection because they're either in their nest, they're underground, um, it's winter, so they're overwintering, or, it's raining, so they're not out. Um, or maybe some other thing. Maybe it's a cold snap. There's all kinds of factors that can influence whether or not you're going to actually detect a species, even if it is present at your site. And so, do I want to try the Mentimeter again? I do have a Mentimeter for this question, but I don't know if it's worth, well, actually, let's see, because now I think you might be able to see my screen. Tell me if you can once I get there. Oh, there's your beautiful Mentimeter. I cut it off before all the responses were there. All right.
We can see it.
You can. Okay, great. Let me make sure I get on the right one. Okay. Go ahead and try this one. So there's the code. It's actually the same code. So hopefully it just popped up automatically. And so biotic just means living and abiotic is non-living. So what kinds of factors do you think might affect whether or not you detect a bee at a site? And while you're typing, I'll tell you about that picture because we saw some Fervidus pictures earlier. That's just a Bombus fervidus worker descending down into her nest. And that's a surface nest. So it's kind of under a clump of grass, which Bombus fervidus, at least in the Midwest, I have no clue about the Bombus fervidus out here, but they are known to be surface nesters. Awesome. So let's look at some of these while they're still coming in, because I'm also realizing I'm going way too slow, so I want to speed it up a little bit. So we've got weather and temperature are big ones. Flowers, I see things like light and soil. I see lots of flower stuff, floral things, time of year, habitat, tree height. That one, I would love to know more about that because that one's really interesting to me. Pesticides, accessibility, training of observers. Awesome. So we've got a lot of great answers there. I'm going to go ahead and go back over to my slides, but feel free to keep answering it. So I'm going to save these later. So I have a few helpful detection terms before we move on, just to make sure we're all on the same page. So imperfect detection, we kind of got at that already. It's when a species might be at a site, but we just miss it. We didn't see it. Just like our spiders in the room. Detection probability, it's the probability of detecting a species at a site if it's there. Of course, if the species truly isn't there, you'll never detect it. So detection probability in that case is 0. But in general, detection is going to be somewhere between 0 and 1. 1 would be perfect detection, which just isn't, it might work for dogs in your apartment, but for bees, I don't know that there's ever a site or a time or place where there's perfect detection. And then building off that, cumulative detection probability is the probability of detecting a species at a site, but across multiple surveys. So let's say you went out once a week for a month. So then it's what's the probability of detecting it across all of those. So I'm going to get into some of my dissertation research, which focused on this concept. And I really wanted to know what are those factors that affect bumblebee detection, more specifically for rusty patch bumblebee, but I studied the entire community, bumblebee community in my area. I also wanted to know how detection probability varied between stable versus declining species, basically to know, are you more likely to detect something common than something rare, if it's indeed at a site, and how many surveys should be conducted and when to detect imperiled bumblebees. So I'm going to just share some results from this paper, but you can always look it up. And I'm not going to talk too much. It's a lot about occupancy modeling and it's kind of dense. So I didn't think that would be very fun for an evening talk. So I'm not going to talk about occupancy modeling at all. But if you're curious, you can go read that paper. So for this study in 2018, we just focused on the 7 metro county area of Minnesota. So pretty restricted. And we selected 94 random sites along roadsides. Why roadsides? The honest answer is because we were funded by the Department of Transportation. But I'd say the science approved answer, because it's never enough to say it was funding, that roadsides can be used for restoration. It represents a large area of land that's available for restoration. Though I would say I'm mixed on whether I think it's really great to put in pollinator habitat along roads or not, because it also can lead to road mortality from car collisions, but not what this talks about. But I wanted to put that out there because, a lot of science and a lot of what gets published is really dependent on the funding source. So it's just good to be aware. So we did our field work from June through August, so summer of 2018. And we walked these 250-meter, we call them meandering transects. Basically, it was a 250-meter transect, but we didn't restrict ourselves to walk in a straight line. We wanted to be allowed to walk kind of meandering to where the flowers were. We sampled between 8:30 and 4:30, standard work day. We had 1 observer per survey, but there were 3 observers in the study. So for each survey, it was 1 of 3 possible observers. And we visited each site between 3 and 15 times during the summer. And sites where we found a declining species, we did more sampling there because we wanted to collect some extra data for those species. We also surveyed vegetation along our transect. So all of that is to say we did 561 total surveys. For our methods, they're pretty simple. We walked our transect, we netted, chilled, photographed, released. And we did have to take pretty detailed photos. Actually, in Minnesota and the Twin Cities, a lot of lethal bee sampling is restricted because of rusty patch bumblebees. So we weren't allowed to do any lethal sampling. So we had to make sure that we were getting pictures, depending on the species of, I call it the armpit, but it's kind of just the side of the thorax. Sometimes the face was important. I think someone mentioned long and short faces, and this isn't the best picture because they're not the same scale. But so Rufocinctus, for example, has a short face. So the distance between the bottom of the eye and the mandibles is short compared to versus Bombus vagans, which has can have similar color pattern. Rufocinctus is variable, but it has a long face. So it was important to get the face, the sides, the backs, and the thorax and the abdomen to be able to correctly identify these species. So in this case, the factors that we wanted to use in our model to investigate, we selected them ahead of time based on published literature and expert opinion. So we wanted to see how date, time of day, and observer experience level mattered. So we had the 3 observers, and so left is me, and from left to right is lowest to highest experience. So in this study, this was my first year of grad school. I was actually the least experienced of the observers. So we went out, we did our surveys. We found just over 5,000 bumblebees from 15 species. From the photographs, we did struggle to separate some species. So there's a couple that are species groups where we just lump 2 species together. And everything above that red line is what was analyzed in our models. So that's what the results are that I'll show. So for our first question, I wanted to know how detection probability varies between stable and declining species. So on the left are the stable species and the right are the declining species. And those are the mean detection probabilities. So the probability of finding that species at a site if it's there. And we were surprised to see that Bombus affinis, it's right in the middle, really. The stable species varied from 33 to 73%. And Bombus affinis, it wasn't markedly lower, it fit right in. But one thing to note is these are mean detection probabilities. So during the mean sampling date time of day and for observer 1. And that's important, and we'll show, because these things are dynamic, actually mean detection isn't the most useful thing. So here's what detectability looks like for time of day. So on our x-axis, we have time of day between 8 and 4:30. And then we have detection probability between 0 and 1 on the y-axis. And you can see that detection increased ever so slightly as you got later in the day, but the relationship is weak. So really, I would say there's really no pattern here, nothing too discernible. That wasn't the same for date. Date was very important. And so we included date in both a linear and quadratic form in our model. And we saw that species varied in when their detection dates peaked. And I used ordinal date there, but essentially kind of midsummer July is where a lot of these species started to peak. But they are a little different. So like Bombus bimaculatus, for example, peaked earlier. It's an early emerging species, so that's not surprising. Bombus impatiens, which is or Bombus fervidus, sorry, which is a declining species, reached its peak detection much later in the season. And so we used this data, our detection data, to calculate the cumulative detection probability. And just a reminder, that's the likelihood of detecting a species across multiple surveys. So now on the x-axis, we just have number of surveys. And so to answer this question of how much sampling effort you need, it really depends on when you're doing surveys. So for example, if you did 4 of these 15-minute surveys on these different dates, and we just picked dates kind of at the beginning, middle, and end of the sampling season for this example. So earlier on in the season, early July, you have a really good chance of detecting Bombus bimaculatus, but by August, that's tapered off significantly. You're much less likely to find that species. And of course, that's related to its phenology. But Bombus affinis, our target species, shows a different pattern where it's, a little bit lower at the beginning of the season, it peaks in the middle, and then it starts to taper off slightly. But then we saw a different pattern with Bombus fervidus, where detectability was pretty low at the beginning of the season, but it just kept increasing until the end of the season. And so if your goal, say, was I just want to sample the whole bumblebee community, based on these numbers, I would say, sample more towards the middle of the season. It's not the highest detection for Bombus bimaculatus, but it's still pretty dang high. Whereas if you're doing like a more targeted survey, let's say I want to find Bombus fervidus, I would suggest, put your effort more towards the end of the season. So that's kind of how this data and this concept can be used to guide monitoring and sampling. And so the key takeaways from that first study are that rare species are not necessarily less detectable than more common ones, meaning if they're there, you can find them. Certainly, if there's a lower density of a species, it's going to be harder to find, but we didn't see any strong patterns, at least in this bumblebee community, suggesting that rusty patch bumblebee were, much less detectable than any other species. And so bumblebee monitoring programs can use information like this to optimize their detection. And in fact, the rusty patch bumblebee long-term monitoring, like federal monitoring program, is just rolling out this year. So I'm excited to see how the first year goes. Excuse me. And another key piece is aligning your cumulative detection probabilities with program goals can help sampling for rare species. So that was what I was getting at with that last slide, where depending what you're looking for should really guide when you're going to go out and survey to optimize resource use. So that was the first chapter of my dissertation. I'm going to share next some information from the second chapter. And this one, really the main objective of this study wasn't detection. This time it was actually about, occupancy. So presence, absence of rusty patch, but we're not going to talk about that. But if you want to know, you can read the paper. I'm just going to focus on the detection piece of that study. So building off of what we did in 2018, we wanted to further analyze associations between those different factors and detection probability. So the first time around in 2018, we picked our factors ahead of time based on expert opinion. But after seeing especially that time of day result where, there really was no pattern, I thought we just don't know enough to rely on expert opinion at this point. So this time I wanted to be more systematic. So one thing I did was expanded my search rather than only focusing in on the Twin Cities where we know there's lots of rusty patch bumblebees. I tried to go out into other areas where it might occur. So I'll point out on the map, box B, that map was updated in 2023, and there actually used to be priority area in that box, but it went away. It was updated. But I had already collected the data, so no going back. This time I sampled in restored prairies, so I wanted to sample only in high quality habitat rather than roadsides. Because these are more geographically separated, we could only get to 24 sites. So it was a trade-off where we had to have fewer sites, and we stratified our sampling by 3 different habitat types. So natural, agricultural, and developed areas. But I'll point out within those, so that's like the surrounding landscape, we did our actual surveys in restored prairies. So these sites were not completely randomized like in our first study, which, again, is a trade-off. But we really wanted to focus on areas in those priority zones. So again, we sampled basically the summer. Summers in Minnesota are short, so you only have a couple months. Pretty much everything else, well, there were some other differences. So we did 60-minute surveys across 3-acre areas. So we covered a larger area and went out longer. Instead of doing the replicate surveys where we did more effort at sites with rare species, we kept it even. So just 5 surveys at each site. So we only did 120 surveys this summer. But everything was done a lot more consistently and standardized because I wanted to really get at those detection probabilities. So this time I collected covariate data for 10 different things: temperature, cloud cover, wind speed, time of day, date, floral cover. But floral cover, it's actually a category of floral cover, and I'll show you that later. Number of native blooming plants, Bombus to Apis ratio. So that's an interesting one, but it's directly from the Fish and Wildlife Survey protocol. So you do your survey and you come up with these estimates of how many bumblebees to honeybees, latitude, and then rusty patch bumblebee preferred plants. So the Fish and Wildlife Service has published a list of rusty patch bumblebee preferred plants. So this was just a simple presence absence. Either one of those plants is blooming at the site or it's not. So this time we found, by expanding the search, we did find a lot more bumblebees, about 7,600 of 16 species. We ended up including 7 species in our models. And my slides it's kind of weird. The ones we included are actually by these icons. So we did get rusty patch. This time we did separate Furvitus and Borealis, and then we had a couple other ones in there. So our top 6 detection covariates in order were date. Well, date and floral cover were tied, really. Then that rusty patch bumblebee, number of native plants, wind speed, and temperature. And I think all of these things came up in our word cloud. The numbers in parentheses, that's how many out of 7 species where it was a strong relationship. So you can see after wind speed, it drops off. So temperature was only really had a strong influence on 2 species. But wind was one that I guess I just wasn't expecting because prior to that, I had sampled more in the Twin Cities area, but especially out in western Minnesota, which is historic prairie and very windy, wind speed had a really big impact on what was detectable. So here's very similar to the first study I showed you, just showing date and showing the different detection curves. And 6 out of the 7 species reached their peak detection within a 10-day period between July 19th and 29th. So that early species, Bombus bimaculatus, we missed that one. But if you were designing a monitoring program in this area, I might suggest focusing kind of on that last week of July. And so here's those ordinal floral cover categories. So it was 1 through 5 based on the observers' estimate of the percent of area in the 3 acres covered by blooming flowers. So it's pretty rough. We didn't have time to do the detailed vegetation surveys, but even with this rough measure, all 7 species, of course, as you know, and showed up in our word cloud, had a positive association, or they were more detectable when there was more floral cover. Does this talk go till 8:30?
Yes, yep, till
Oh
8:30.
goodness.
I realized we ran a little over, so if you're comfortable with running over and people would like to stay, happy to catch up on those couple of minutes if that's helpful.
Okay, I will speed it up, but yes, and if folks have to jump off, that's understandable too. So what did we learn? Date and floral cover are super important factors for detecting bumblebees, but I'm going to speed up too. Bumblebees in our study system are also more detectable at sites with specific preferred plant species and with many native plants. And as I said, wind speed also strongly influenced detection at Western sites. So that's the studies I did. I did want to point out though, detection probabilities, they're not transferable across the entire range. I would maybe hazard to say maybe you could extrapolate some of this to the rest of the Great Lakes population, but that Appalachian population, I'm sure, would have very different factors that were important for Rusty Patch and probably would peak at different dates. We also just published a study on the range-wide genetics of Rusty Patch and found that the Appalachian and the Great Lakes populations appear to be 2 distinct genetic populations. So there's a lot going on. There's a couple papers there if you're interested. You could check those out. And so I wanted to make this, about the West. So I grabbed a couple studies I'll just share really quickly about Western bumblebee. So as was mentioned already today, this used to be a really common species, but it's declined over 90% since the late 1990s. It declined. It was actually petitioned for listing, like I said, in 2015, and we're finally expecting that listing decision. And so previously, Western bumblebees were reared commercially back in the 90s. But pathogen loads of this specific fungal parasite got so high that it just wasn't viable to keep these colonies going. And so commercial rearing operations abandoned attempting to rear this. However, in conjunction with these higher pathogen loads of the commercial species, there was also a decline in the wild bees. And it's widely believed that pathogen spillover from those commercial bees that were being reared, probably it probably got into the wild populations and contributed to the decline, among other things, right? There's lots of factors involved in bumblebee decline. There's climate change, there's pesticides, there's habitat loss, there's all these things, but it's very likely that these commercial rearing operations also contributed to its decline. So there are a few studies that looked at detection probability for Western bumblebee, so I'll just show the results quickly. So this first one is by Graves et al., and they analyzed a long-term data set. The data is from 1998 to 2018. And in their model, they used land cover, bloom date, which is really interesting, days since bloom initiation, and then an interaction between site-specific bloom date and days since bloom. So here's what they found for Western bumblebee detection. Let's focus on the graph on the right. So you can see the detection of the species peaked around the same time every year, but detection probability got much higher around peak when bloom dates were earlier, so that March 1st bloom date, versus if bloom date, initial bloom date was in May or July, detection just never got as high, probably because there were fewer colonies. I'm sure having those initial spring flowers helped more colonies to thrive and make it that long. And then the second study I'll show, this one's by Janicek from 2023, so it's really new. They analyzed 23 years of data, and they were actually looking to assess the effects of climate change, land cover, and pesticides on occupancy. But this is a detection probability talk, so we're not going to look at that. For detection, they used growing degree days, which is just a tool, basically a measure of accumulated heat, and it's used for monitoring growth of plants and insects. And they also use percent forest cover. Let's skip that one. So detection probability in this case for Western bumblebee, very similar. This time instead of date, they have growing degree days, but they still found, you know, in the middle of this, the growing season is when detection peaked for this species. They also found that detection peaked as the percent of forest increased up until 52%. And at that point, the mean decreases, but you can see that shaded part, that's the error. It gets so wide at the end, you really can't say much about it, but at least initially detection increased as forest cover increased. So let's skip that one because we're running out of time. So what can we learn from these studies? Well, as we've seen for both rusty patch and Western bumblebees, and you know, bees generally, date and floral resources are really important factors that influence detection probability. But they're complicated because it's not just plant cover or number of plants. It also matters things like bloom initiation, the phenology of the plants, but all different metrics associated with those floral resources affect detectability. But it's also important to know most studies that have looked into detection probability of bees have focused on bumblebees. They're large, they're easier to see, and their flight period is a lot longer than most bees. And so I'm not sure how much of these studies really apply directly to non-bombus species. That's unclear at this point. But repeat sampling is really key. As I've mentioned, bees can't really be detected perfectly. Oh, bees cannot be perfectly detected in any given survey. Okay, I did say it right in there. So doing repeat surveys is really important. And if you're looking at a bee or you're interested in a bee that only has, you know, a 2-week flight period, that just means your repeat surveys may need to be fairly close together. And so these are my last takeaways. The observation process is critically important. Information on how different biotic and abiotic processes influence detectability can actually help us improve our sampling efficiency and improve monitoring efforts. And for species that lack information on detectability, which is most of them, nearly all of them, we can still consider, based on what we know of bees, what might be important factors. And we can use what we know combined with repeat sampling to increase our cumulative detection. And hopefully researchers will take on some studies of those harder to study species. So I can also send these out later, but these are the citations for the studies I highlighted. My acknowledgements, of course, we worked with lots of partners. I had to get lots of land access permissions to do this work. Of course, I have to mention my funding, fellowships and grants that supported this work.